Hello, my name is Dr. Roderick L. Rowe, and today we are going to go over the answers for the experiment on blood typing. So in lab, you took this white tray that's over to the far right, and you put drops of a patient's blood, and it was a fake simulation of how it would really be done if you had pricked a finger on a patient. So let's look at the table for patient one. Patient one, we had agglutination occur for antigen A, not for antigen B, but just for antigen A. So let's just look at patient one. So now you go down and look at the image, and this is how it would actually appear if we were able to use a high-powered microscope to see the cell membrane. So on the cell membrane of a red blood cell, you would have this this molecule that would be antigen A. Now, so if the antigen reacts with the antibody that we're going to use, then you will have an agglutination occur. Agglutination is this clumping of antigen and antibody. They both hybridize, they both are attracted, they both have this affinity to one another. So you added the drops of your fake antibody, it bound to the antigen A on the molecule in the white plate. That means this patient has blood type A. So I explain it in the sentence in the red. So if the reaction agglutinates for antigen A, the red blood cell has antigen A. If that is a true statement, then the patient has blood type A. Here's the second one. This is for patient 2. You did the same experiment, but now patient 2, you had agglutination occur when you use the antibody B. That means, look at that last statement, if the reaction agglutinates for B antigen, the red blood cell has B antigen. So the red blood cell has that big old B on the cell membrane. You add antibody B and it agglutinates. So if B agglutination is a positive reaction, that patient has blood type B. The next patient is patient 3. You use that tray. You saw agglutination for A and B. What that means is that the red blood cell surface has antigen A and it has antigen B. Therefore, you had agglutination of both. So let's read our last statement. If the reaction agglutinates for A and B antigen, the red blood cell has both antigen A and antigen B on its red blood cell receptor on the red blood cell membrane. That patient has AB blood type. So notice that this patient doesn't have any antibodies for either A or B. So they can receive any blood type because they won't have an agglutination reaction. They can receive a blood transfusion from a person with A blood. They can receive a blood transfusion from a person with B blood. And definitely they can receive one from a person with O blood. The last one, this person was patient 4, and they didn't exhibit any agglutination. So that means their red blood cell didn't have any markers on it that were foreign, that were antigens. So if the reaction does not agglutinate for A or B, they didn't have any antigens. So the red blood cell does not have any antigen A or antigen B. This patient has blood type O. Since there are no antigens on that cell, they can be the universal donor. Let's really quickly look at B lymphocytes. The purpose of this simulated blood experiment was not to determine what blood type we have. It was to determine how B lymphocytes and T lymphocytes work in our immune response. So let's look at the B lymphocyte or the humoral response. And we're going to use the same stuff we just went over. So patient 1 was determined to have blood type A. So if we're looking at our picture, if blood type A has an antigen A in that patient's bloodstream, they would have antibodies against B blood. They don't produce B blood, so they would have antibodies against it. So remember, we talked about a naive B cell. So this B lymphocyte, which is born in the bone marrow, then matures in the bone marrow. It will grow these receptors on the surface of the B lymphocyte. That receptor is primed for a specific encounter. And remember, these encounters are things that are foreign to that patient. So that patient has blood type A, so what would be foreign to them would be to get a different blood type, like B. So therefore they have an antibody specifically for blood type B. So B lymphocyte is primed for that encounter. It is a virgin. It is naive. If it is ever exposed, then agglutination will occur. So let's say the patient receives a blood transfusion. But this blood transfusion was an accident, and they gave type B to a person with blood type A. Immediately agglutination would occur. That looks like the image to the right, where antibodies will grab and stick onto the antigen or the epitope on this red blood cell and make it clump together, just like it did in your small white little glass petri dish. So that is how this blood typing experiment relates to the actual lecture that we're discussing, where we're looking at how the humoral response benefits us in reference to our immune system. We will now look at T lymphocytes. We talked about T lymphocytes in lecture, but how does it relate to this lab? Well, T lymphocytes are part of the cell-mediated response. There's going to be two types of cells that I'll mention, the T cytotoxic cell and the T helper cell. They are cell-mediated responses. So we're going to use the same experiment. Let's stick with patient 1. Patient 1, we determined that they have A blood type. Use the same picture. They have an antigen on the surface of the red blood cell that is antigen A. Patient 1 has a naive T lymphocyte receptor that is primed for an encounter with B antigen, right? So The T lymphocyte starts off, it is born in the bone marrow. Then it moves to the thymus gland where it will develop its receptors. And it's going to develop receptors for everything that is foreign to it, to that patient. So this patient has antigen A, so the T lymphocyte receptor will be for B antigen. So if you ever give this patient a transfusion of B red blood cells, an antigen presenting cell like a macrophage or dendritic cell will gobble up that red blood cell, process its antigen, and expose a piece, which would be the epitope, on its surface. And its surface would be either one hand would be the CD8 receptor, or the other hand would be the CD4 receptor. If it is the CD8 receptor, it is going to then bind to the T cytotoxic lymphocyte. At the same time, remember that antigen presenting cell can have two hands. So that same antigen presenting cell can present on another site, such as CD4, that specific epitope also. Now, if it's CD4, then that's going to bind to the T helper lymphocyte. So you can see how a transfusion of B blood type will then activate the T cytotoxic lymphocyte and the T helper lymphocyte simultaneously. Now, both of these signals the release of interleukin 2. And interleukin 2 is going to lead to clonal expansion of both T memory cells and T cytotoxic cells. So basically, that interaction, that immunological synapses that was just created between this antigen presenting cell to stimulate both T helper and T cytotoxic is going to lead to the attack of the clones, where we have that one T cytotoxic cell making thousands of T cytotoxic cells. And then now they're going to go try to target all of those red blood cells that are foreign. The red blood cell type B will then be ruptured when granzyme B and porphyrins come in and destroy those cells. So I just wanted to make sure you understand the purpose of us doing this red blood cell, this ABO blood typing. It was to help you associate our T cell mediated immune response and our humoral B cell response. Then finally, you should make sure you know how to do the RH agglutination portion of it. It's the same concept. If it agglutinates for RH antigen, the red blood cell has the RH antigen, then that patient will be RH positive. So if you understand how to do A, B, and O blood typing, this RH factor or antigen is very similar. It's just a different molecule. Finally, we'll look at one of the tables in your lab, which was the patient antibody production. What type of antibodies the patient will produce. This is pretty simple because the answer was in a previous image. So for example, patient 1, you have determined that they have an antigen that is named antigen A. If they have antigen A on their red blood cell, they would produce antibodies against the foreign red blood cell, which would be antigen B. So then this person has antibodies, antibody B. So this person has antibodies for B blood type. So then you would just put the positive with the antibody. Then if you follow along, now you know what antigen it has, because on this picture it's telling you what antigen it has. So that is pretty easy table to figure out. Then finally, this is your key. So make sure your answers align with this.